This video is going to consider cell structure and what we can see beyond the light microscope. So when you've been looking down the microscope at cells that you have mounted yourself, like onion or algae, or looking at banana cells, you can see some features. You can see cell wall, cell membrane, you can see a nucleus, you can see cytoplasm, and you can see chloroplasts. But as you can see from this image with an electron microscope, cells are much more complicated than that. And inside the gel, which we sometimes call cytoplasm, there actually are a large number of complex compartments called organelles. So organelle is the word given to a little organ, and that's literally what the word means, and we are talking about the substructure within a cell. So let's have a look at the different parts. So when you look at a nucleus under an electron microscope, you see that it actually has an interesting structure based on essentially a circle, a bag, membrane-bound bag. So it's got a nuclear envelope surrounding the DNA. So you can see the nuclear envelope here. And it's a double membrane, but you'll see there are little holes in the membrane. So there's a bit of a gap there. And over here, there's a gap between outside and inside, and this is important, there's one there, for allowing the messenger RNA, which is going to carry the message from the DNA out into the cytoplasm to make proteins. So we've got pores, we've got DNA inside, which stains quite darkly in different places, depending on how active the DNA is there. And that's our nucleus in the middle of the cell, a really obvious structure to see. Next, we'll have a look at some more membrane structures. So the endoplasmic reticulum. Just orientate you a little bit here in this falsely coloured stained electron micrograph. This is the edge of the nucleus here, and here is a mitochondria up here in purple. So we've got here all these membrane structures where you can see the inside of the membrane is green, yellowy green, and on the outside there are little red dots. The red dots on this are the ribosomes, and they are stuck to the outside of this endoplasmic reticulum membrane, and because they're involved in protein synthesis. Sometimes you get membrane structures which do not have ribosomes attached. That is smooth endoplasmic reticulum, as opposed to the rough endoplasmic reticulum with the ribosomes attached. So the smooth endoplasmic reticulum isn't doing protein synthesis because it hasn't got any ribosomes. It's involved in either making or breaking up lipids or fats. So you'll see lots of endoplasmic reticulum in metabolically active cells that are building proteins or building fats or breaking down fats. Also part of the membrane systems in the cell, we've got the Golgi Apparatus. This is also membranes. They're stacked on top of each other. So you can see a sort of cartoon version of them on the right. And these stacks are moving all the time. They're all budding off one side and joining onto the next. You see little vesicles associated with the pictures in the electron micrograph. See all these little vesicles here. And what we've got happening is that it's attached to the endoplasmic reticulum on one side, and then slowly, as processing of the proteins happen, it moves through the stacks from one stack to the next stack, and eventually, if the protein is going to be leaving the cell, these vesicles bud off the ends of the stacks and they make their way to the surface of the cell. So this is involved, you see lots of Golgi if you've got a cell that is secreting proteins, like maybe a salivary gland cell. It's making lots of proteinaceous saliva. So that's where you would see a lot of Golgi apparatus in a cell that's doing that. And here's a picture of it happening a bit more cartoon-like. So we've got the endoplasmic reticulum in sort of orangey pink, and you've got vesicles budding off the side, going into the Golgi apparatus, which is the dark pink, being processed, and then being sent to the cell surface to go and do whatever job they need to do outside the cell. Lysosomes are another vesicle. They originate from the Golgi, and their job is to be a bag of enzymes which will either destroy something that's been engulfed by the cell, or they will maybe destroy a worn-out organelle if an organelle has reached the end of its life. In the electron micrograph, this is a cell which is a white blood cell, and its job is to gobble up bacteria and take them inside. So you can see all of these big lysosome structures. There's lots of them because that is the job of this cell, to destroy foreign bacteria that this cell has engulfed and gobbled up. So next structure we've got is not connected to all these membranes. It's a structure all of its own. It's called a mitochondrion. Many of them would be mitochondria. So lots of them in every cell, and if it's a metabolically active cell, there will be even more, because this is the site of aerobic respiration. It's where we make ATP, which is the energy currency of the cell, and it's what we then use to move, do secretions, whatever the active cell needs to do. So in this we see a double membrane layer. So we've got an inner membrane and an outer membrane. So you can just about see that on this image here. So we've got an outer membrane, and then on the inside there an inner membrane. It's a double membrane structure. And the inner membrane is folded backwards and forwards to create a really large surface area for all of the reactions of respiration to take place on. And those inner foldings are called cristae. So you can see them here on this cartoon representation. So folding in and out on that inner membrane, giving lots of surface area for reactions to take place. So remember, if you see a cell with lots of mitochondria, you will know it is a very metabolically active cell. Here is another very similar shaped organelle to the mitochondria. It also has a double membrane, and this one is chloroplast. Chloroplasts are only found in photosynthesizing cells, so generally a plant cell, and they have the double membrane structure again on the outside, and inside, instead of having a folded membrane, they have stacks of membranes. It looks like coins have been stacked one on top of the other. And that little stack is called a granum, and the actual membranes are called thylakoid membranes. And that's where photosynthesis takes place, actually on those membranes. So in this big picture of a whole plant cell down here, you can see several chloroplasts. So here's 1, 2, 3, just half of one here, 4, and then 5, 6, and another half one here, 7. So we've got several chloroplasts. You can compare their size to the nucleus. Here is the nucleus, the kind of grey speckled area, and you'll also notice a big vacuole in the center. We'll come to the vacuole in a moment on the next slide. So that's a plant cell with lots of chloroplasts with a very classic internal structure to them that's different to a mitochondria. So here is the vacuole again. I've got an electron micrograph picture of lots of different cells so that you can clearly see vacuoles in many different cells. A large space, a void, there's not a lot happening in them. They are just a storage for water and waterborne storage chemicals, and you can see that because of the vacuole, all the contents are pushed out to the edge of the cell. So that's a really classic feature to look for in plant cells, because that's where you get really large vacuoles. And they've got a membrane surrounding them to hold all the sap in and hold the rest of the cytoplasm out. Cell wall is also unique to plant cells, but there are different types of cell walls also in fungi and in bacteria, but you don't find them in animal cells. The area which is a cell wall in this picture is the area in the center here between the cells. So we've got a cell membrane on this cell here and a cell membrane on this cell over here, and then you've got their cell walls in between that are essentially contacting each other, and you can't really distinguish one from the other very clearly in this picture. But cell walls in plants made of cellulose and in different materials in different kingdoms. Now, there are other parts to the inside of the cell which are also there, but you may or may not need to talk about them. We've got here stained with fluorescence markers because the electron micrograph pictures don't pick them up very well, but we can stain them with a fluorescence marker using fluorescence microscopy, and you can see a very complex internal skeleton in the cell. It's made up of microtubules, and it's used for structure support and to change the shape of the cell or to move organelles around inside the cell. And it's only really fairly recently that we realized that this was the complexity inside the cell, thanks to the development of fluorescence markers and fluorescence microscopy. The centriole is associated with this because the centriole is a very tiny organelle, very close to the nucleus, and it helps to organize all the tubules to help pull apart chromosomes correctly during cell division. So you might see them in a micrograph picture, but they are very small, but we will talk about them more when we think about mitosis and meiosis and cell division. And the final structure we'll think about is a structure on the surface of a cell, little hair-like structures. If they're short, they're called cilia, and if they're long and they whip around, they're called flagellum. And these are structures that are used to either propel a cell along or to move anything that's contacting the cell past the cell membrane. So these again are structures that you'll see, and the electron micrograph used to make these images is a scanning electron micrograph because you're actually getting a 3D image of the structure. So as you can see, it's much more complicated than we can see down the light microscope, and thanks to all the advances in technology that we have, we've got much better resolution to get in and see more detail, and a much better magnification so that we can see much smaller structures like cell membranes and ribosomes and the like.